Hello students, now we will start with new topic that is about other type of biomolecule in biomolecules chapter, that is nucleic acids. So nucleic acids are nothing but DNA and RNA. If we come to definition of nucleic acids, these are formed from nucleotides, or nucleic acids are a combinations of nucleotides, are genetic material formed by fusion of nucleotides. We can also say many number of nucleotides constitute into nucleic acids. So what are nucleotides composed of? These are composed of pentose sugars plus nitrogen base plus phosphate molecule. What type of pentose sugars are involved in nucleotides or nucleic acids? Pentose sugars are of two types. What are those? In previous carbohydrates topic we have discussed. One is ribose sugar. Second one, deoxyribose sugar. Ribose sugar is seen in RNA. Deoxyribose sugar is in DNA. Then what about nitrogen bases? Nitrogen bases again are of two types like that of pentose sugar. One is purine types, another one pyramidals. Now, one phosphate molecule will be involved in the reaction. Its basic structure will be like this in case of nucleic acids. Because of that, this learning of this phosphate structure like this helps us to learn about number of ester bonds and how they are formed, and number of diester bonds that are present in the nucleic acids like DNA or RNA. Now, coming to purines. Purines are made of two rings, benzene ring-like structures. Two rings, so-called dicyclic type of rings or dicyclic type of substrates. So is dicyclic. Now, purines are, what are examples of purines? Are adenine and guanine. Now, purines have nitrogen atom at 1, 3, 7, 9 positions of carbon. Positions of the rings. If you see the typical structure of a purine, it will be like this. We will be giving the numbering like 5, 6, 7, 8, 9, 4, 3, 1, 2, 3, 4, 5, 6. We will give the positions like, in case of purines, the numbering like 1, 2, 3, 4, 5, 6. Okay. Now, I have written here one point. Nitrogen atoms are at 1, 3. So, here will be nitrogen at first carbon position, another nitrogen, and another nitrogen. Now, it differs from adenine and guanine. What differs? Other functional groups that are present in case of purines. This first ring is called as benzene ring. Benzene ring. And this second ring is called as imidazole ring. Imidazole ring means two types of rings are involved for formation of one compound, that is purine. Now, as I have said you, one ring is benzene ring, another ring is imidazole ring. The positions must be given as 1 carbon, 2nd, 3rd, 4th, 5th and 6th, 7th, 8th and 9th positions. In case of adenine, at 6th carbon, there is amino group, and at 1st, 3rd, 7th, 9th, there will be nitrogen atoms. I am not mentioning here all the double bonds, etc. perfectly balancing, because we in biology, we need just what are the functional groups that are present in adenine or guanine, like that of purines or pyrimidines, etc. So adenine structure is having amino group at its 6th carbon position. Now, next is guanine. Again, we will draw 2 rings since purines are of dicyclic in nature. 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 positions. But here at 6th carbon there is ketone group, and at 2nd carbon there is amino group. In case of adenine, amino group is located at 6th position. In case of guanine, amino group is located at 2nd carbon position. And the existence of nitrogen atoms is again same. So these are purines. Then what about pyrimidines? Pyrimidines are monocyclic, means only one benzene ring will have additional different functional groups at different positions, and they will be converted into pyrimidines like thymine, uracil and cytosine. These are monocyclic. Nitrogen atom is at 1st and 3rd carbon positions. So if we write here, what are examples under pyrimidines? Thymine, cytosine and uracil. This uracil is existed in case of RNA, in RNA instead of thymine. This thymine is seen only in DNA. And it is absent in RNA. So we say one important point, that is total nitrogen bases. We represent nitrogen bases like this, capital N, small b. Nitrogen bases are 5. What are those? Adenine, guanine, cytosine, thymine, uracil. Next, nitrogen bases of DNA are adenine, guanine, cytosine, thymine. Nitrogen bases of RNA, adenine, guanine, cytosine, uracil. Coming to structures of thymine first. It is having ketone group at one position. First of all, we have to give numbering here. So we'll give 1, 2, 3, 4, 5, 6. So 6 carbon benzene ring is present. At 1st carbon position there is nitrogen occupied. 3rd carbon position also one nitrogen. So it is having one ketone group at 4th position and another ketone group at 2nd position also. And it also has one more functional group, that is methane, at 5th carbon position. Next, coming to cytosine, or we will write cytosine. What is its structure looking like? Again benzene ring. I am giving carbon numbering as 1, 2, 3, 4, 5 and 6. At 1st carbon there is nitrogen. 3rd carbon position there is nitrogen. At 4th carbon, in case of cytosine, there is amino group like that of adenine. But adenine is having amino group at 6th position. That is ANH2 it is having. Now, at 2nd position of cytosine, again there is one ketone group. So this is the structure of a cytosine. Coming to uracil. Uracil is the pyrimidine that is seen only in RNA, not in DNA. So its structure is Again benzene ring, carbon positions 1, 2, 3, 4, 5, 6. Nitrogen atom, your nitrogen atom. It is having one ketone group at 4th carbon position, another ketone group at 2nd carbon position. If we match the thymine and uracil, there will be some similarities. Like ketone groups are present at 4th position and 2nd position. Here also in case of uracil the same thing. But the only extra point in case of thymine is it is having one extra methyl group at 5th carbon position, but that is absent in case of uracil. So we can say this thymine with another name as methylated uracil or 5-methylated uracil. Means 5 means 5 carbon position. 5-methylated uracil. We can also say other term for thymine as 5-methylated uracil because complete biochemical structure is the same or similar to that of uracil, but thymine is having only one extra methyl group at its 5th carbon position. So this are attached with pyrimidines with the help of hydrogen bonds. Example, if we see adenine, a purine, is having double bond when it is binding with thymine. Means one purine will be binded with one pyrimidine only. Adenine always binds with thymine only. This one simply we represent like this, AT. Next, guanine, another purine, is bonded with another pyrimidine, that is cytosine, with 3 hydrogen bonds. This we represent simply like this. So these two conditions in DNA. Then what about the binding of adenine with uracil? Adenine is again with 2 hydrogen bonds or double bonds. It is binded with uracil. This one in case of RNA. And here we represent like this only. In the same case, guanine, it is same. Guanine triple bond with cytosine. This one also in case of RNA, it is same. Next, in the nucleic acids or nucleotides. In the nucleic acids, purines are binded with pentose sugars, or we'll write nucleotides, we'll write so that it will be easy. Nucleotides. The purines are binded with the pentose sugars at first carbon of pentose sugar with 9th nitrogen atom position of a purine. Then how pyrimidines are binded? I am going to erase this one. Pyrimidines bind to pentose sugars at first carbon of pentose sugar and first nitrogen atom of pyrimidine base. That can be either thymine or cytosine or uracil, whatever it can be. So this is how purines and pyrimidines are binded. We'll see one example of diagrammatic representation or biochemical structure of a nucleotide of DNA. Then what are the biochemical structures present in nucleotide of DNA? One is deoxyribose sugar plus nitrogen base What are nitrogen bases in DNA? Adenine, guanine, cytosine and thymine. Next, and phosphate molecule. If we frame out the biochemical structure of the nucleotide of DNA, pentose sugar, that is deoxyribose sugar, will be having carbon 1, carbon 2, carbon 3, carbon 4 and branched carbon 5. OH, H, H, H. Next, OH and H. And here is one OH. Here the molecule is CH2OH at 5th carbon. So pentose sugar we have represented. I am writing with pentose sugar in PS. I told you at carbon 1 position it is attached with a nitrogen base. For suppose it can be either adenine or guanine or cytosine or thymine. So for suppose we will take adenine. It is attached from its 9th nitrogen atom attached to 1st carbon position of pentose sugar, and this one 5th carbon position of the pentose sugar, it will be attached to the phosphate molecule like this, OH, OH, and another OH has to be present here, but this hydrogen of the pentose sugar, OH of the CH2OH, that is 5th carbon, that is removed in the form of water and now forms a bond between oxygen and carbon like this. And carbon. We know that the bond between oxygen and carbon can be termed as ester bond. Ester bond. So ester bond in case of nucleotide is formed by the process of dehydration of one water molecule. So here we are seeing pentose sugar, nitrogen base and a phosphate molecule. This can be considered as the typical structure of a nucleotide. Now, one more MCQ. The bond formed between nitrogen base and pentose sugar can be termed as glycosidic bond. Glycosidic bond. We can also learn types of nucleotides. So, adenine plus pentose sugar plus phosphate molecule. This is called as adenyllic acid. Next, another nitrogen base, guanine plus pentose sugar plus phosphate. This is called as guanyllic acid. Cytosine plus pentose sugar again plus phosphate molecule. This is called cytidyllic acid. Next, thymine plus pentose sugar plus phosphate molecule. This is called thymidyllic acid. These all 4 types of acidic forms are seen in case of DNA. Then what about uracil, another pyramid in RNA? Uracil, which is indicated with capital U, plus pentose sugar plus phosphate molecule. This is called as uridylic acid. This is about the nucleotides, the terms nucleotides. Then what about another term, that is nucleosides? Means if we remove the phosphate molecule from a nucleotide, then that combination of pentose sugar and a nitrogen base is called as nucleoside. So adenine plus pentose sugar is called as adenosine. So here we can write pentose sugar plus nitrogen base. Another, guanine plus pentose sugar is guanosine. Cytosine plus pentose sugar is called as cytidine. Thymine plus pentose sugar is called as thymidine. In the same way, in case of uracil, these 4 are in case of DNA. Next, uracil plus pentose sugar, uridine. So So these are nucleotides and nucleosides. We can also simplify this one as nucleotide minus phosphate molecule is equal to nucleoside. Or we can also write like nucleoside plus phosphate molecule is called as nucleotide. Now, this phosphate molecule and pentose sugars are considered as skeletal structures of the nucleic acid or backbone of nucleic acids. Phosphate molecule and pentose sugars are backbone of nucleic acids or skeletal structures of nucleic acids. DNA is having two number of strands that are running anti-parallel to each other. In a sense, for suppose two strands are there like this in case of DNA, one will be moving one side and another will be moving towards the other side. DNA, it exists in helical form. This shape of DNA as if double-stranded helical form is given by Watson and Crick. So if we draw a structure of DNA, here I have given you two different strands. Suppose think that one strand is moving downward, another strand is moving upward, because DNA is having two number of strands which are running anti-parallel to each other. Let's give, for suppose, if this area of the nitrogen base is having pentose sugar 5th carbon, then we represent this one as 5' position. If this area of the DNA is having 3rd carbon of the pentose sugar, then we represent 3'. Means, if this is the pentose sugar, if 3rd carbon is having OH group, if this is present here, then we call it as 3', and at 5th position we have CH2OH. If that area is present here, then we call it as 5'. So if it is 5', then the opposite end we should represent as 3', and for this one as 5' for dark or black colored strand. Now, there are small grooves here and here. These are called as minor grooves. There are very big sized grooves here. These are called as major grooves. The place from one minor groove to another minor groove, or from one major groove to another major groove, or the depression like structure or the folding like structure, either this or this, means between two minors or between two major grooves like or depressions like or foldings like structures, it is termed as one turn of DNA. One turn of DNA is considered to be having 10 base pairs of nitrogen bases. 10 base pairs. Pairs I am using the term because from here, for suppose if you take, it can be adenine, double bond with thymine, guanine, triple bond with cytosine, cytosine, again triple bond with guanine. Next, adenine, double bond with thymine, thymine, double bond with adenine. 1, 2, 3, 4, 5 we have written. Next, cytosine, triple bond with guanine, guanine, triple bond with cytosine, adenine, double bond with thymine, thymine, double bond with adenine. Next, 1, 2, 3, 4, 5, 6, 7, 8, 9. One more we have to write, like we can represent another adenine with a thymine. So totally how many nitrogen base pairs we are telling? One is not a pair. One adenine plus thymine is considered to be a pair. Means how many number of bases we are having? Or 20 nitrogen bases. Nitrogen bases. Now, the diameter from this position to which is symbolic to this position. Like this. If we write, it can be represented as 20 angstroms in diameter. The diameter or the width of these folded complete structures, we can say from here to here also, it can be 20 angstroms, this diameter. What is the length of this one turn? Length is 34 angstroms of one turn of DNA. How can we represent this 20 angstroms? We can show like the phosphate molecule which is present here and the phosphate molecule which is present here. Because nitrogen bases are having phosphate molecules also, the distance between these two on the oppositely running strand can also be considered as diameter of DNA. So these are the measurements that are required for DNA structure. If we go for biochemically arranged total nucleotides in case of DNA strand, there we will see some extra measurements from these 34 angstroms. Because we know that there are 10 nitrogen bases. So between each nitrogen base, this is adenine and guanine, the next one, subsequent one is guanine and cytosine. Between these two, it is very perfectly planned arrangement or length. That is, it can be 3.4 angstroms. Means 10 number of 3.4 we constitute 34 angstroms. If we write the same value in terms of nanometers, then it will be 3.4 nanometers. If we write the value of 3.4 angstroms between two nitrogen base pairs, then it will be 0.34 nanometers. Because sometimes while the questions are being framed, the values or the units are being changed from angstroms to nanometers. Still we need to remember that 34 angstroms or 3.4 nanometers, still the value is same. 3.4 angstroms or 0.34 nanometers, still the value is same. Now we will see about the biochemical structure of double-stranded DNA. As I said you, this is anti-parallelly running strands. So we will write one strand moving upward direction with pentose sugars. Means it represents that this strand is moving toward upward direction, this strand is moving towards downward direction. Since here the end is 5th carbon, we call this strand as 5' end. And complementary, this strand is having third carbon OH group. So this end of the strand as 3' end. Here you see, this is 5' again. Here if you write, this is 3' end again. So if we combine the phosphate molecules, because nucleotides must have pentose sugar and phosphates. Phosphates will be with 5th carbon, where carbon here, and here with phosphate, decrease the size, 5th carbon, oxygen, phosphate. 5th carbon, oxygen, phosphate. This is bonded with phosphate like this, third carbon, which is having OH group. Here also we can write 5th carbon, O, phosphate, oxygen like this. Oxygen, phosphate like this. So this bond is ester bond. This bond is also ester bond. So in a nucleotide, between two successive neighboring pentose sugars, there is diphosphoester bond. This is called diphosphoester bond. Here also the same. Here also the same. Diphosphoester bond. Diphosphoester bond. But separately between third carbon to phosphate, one ester bond. Now, what about nitrogen bases? With the glycosidic bond, if it is adenine, it will be thymine. If it is glycosidic bond guanine, then it will be cytosine. If it is again thymine, it will be 2 hydrogen bonds with adenine and another glycosidic bond. The distance between two nitrogen bases or base pairs in a nucleotide, this distance we can say as 3.4 angstroms or 0.34 nanometers. And the total diameter between phosphate of this phosphate molecule and up to the phosphate molecule here, we can represent as 20 angstroms in diameter. This is the width we are commonly telling when we are drawing the structure of DNA and telling the diameter between the strand is 20 angstroms, means between the phosphate of one end and phosphate of another end. DNA is existed in 5 different forms. So 5 types of DNA are existed in nature. One is B type, next is A type, next is C, D and Z type of DNA. What we have learned just before, Watson and Crick model of double-stranded DNA is B type. So it is having 10 base pairs. A type of DNA is having 11 base pairs. C type of DNA is having 9.33 base pairs. D type of DNA is having 8 base pairs. Z type of DNA is considered to be having 12 base pairs. This is related to the base pair number. Then the direction of coiling of all these types of strands. This bDNA is said as right-handed helical form. aDNA is also considered to be right-handed helical form. cDNA is also right-handed. dDNA is also right-handed. But zDNA is considered to be of left-handed, coiled form. Means bDNA, aDNA, cDNA and dDNA, they show clockwise direction of coiling. Clockwise direction of coiling. zDNA only shows anti-clockwise direction of coiling. So this is about different types and the structures of nucleotides.